everybody. My name is Emily Britton, and I am the Principal Marketing Manager for Food and Natural Products in North America with Waters Corporation. And I'm really happy that I have the opportunity to present to you all today on this topic. What does quality mean to you? Analytical tools for defining a complex botanical. So I have a picture here, courtesy of my iPhone, that I took. And if you look at it, the picture looks all right. It's aesthetically pleasing. And I'm holding some sort of vape device in my hand. Looks pretty sleek, not sure what brand it is at all. You can see color of some sort of product in the cartridge. But despite, maybe a nice looking picture, or maybe even if you extrapolate out and you're in a store and you're looking at products, how do you really know if the quality of the product that you're looking at is good or bad or sufficient? I don't really know. So ultimately, if we ask ourselves if this is a quality product, and that really goes for anything in life that we encounter, we have to consider who's asking the question and what their role is within the supply chain of the market, because quality means something a little bit different to everybody. So what I want to do here is really just take a moment to put ourselves in the shoes of others and think about how do they define quality in the context of cannabis. So perhaps we can first start with the regulators. So the regulators are there to ultimately keep the consumers safe with the products that they are consuming, right? So maybe quality means to a regulator that the products that are on the market comply with regulations, and that the data that were generated to put those products on the market are trustworthy. Regulators may, define quality as, providers or manufacturers having data that are trustworthy, and those validated methods are used by the laboratories that do the testing. And ultimately, high quality to a regulator perhaps means that the products are safe for consumption. But if we go to the laboratory and think about what they, perceive as being high quality, typically laboratories are, hopefully testing the products that come in with a fit for purpose method, using a method that makes sense for the matrix that's being analyzed. Usually that's a sign of quality. And when they're doing the analysis of these different products, typically they would say, all right, all of the analytes, all of the things that I expect that are in this product, I'm aware of what they are and I quantified them accurately. And maybe quality also could mean that there are no unknowns, no mystery peaks or contaminants that might be found within a product. That would be a way that laboratories may define quality. So as we move down the supply chain, now we can think about what the producer defines as quality, the people who manufacture these products. So perhaps to them, it means that the products that they're formulating meet target specifications. They were aiming for a certain percentage of a certain cannabinoid, and they got it, right? Maybe quality can also mean that they source their raw materials from a specific place, whether they're growing products and formulating them themselves, or they're, getting biomass of some sort or ingredients from a manufacturer or provider that they know and trust. Quality can also mean perhaps different manufacturing processes are used. A lot of people, do some sorts of solvent extraction. Some people really try to communicate quality in their products by using, supercritical fluid technology to create their extracts. Ultimately, quality to a producer can ultimately mean that their products are differentiated from the competition and that they have some sort of edge in their products that would make a consumer want to pick their product over somebody else's. So I mentioned the consumer. I would say that's the end of the supply chain, at least in this aspect. And quality, I think, probably means something a little bit different to consumers than it does maybe all the way at the top compared to the regulators. So to consumers, if you imagine yourself being even in a store and you have a selection of products that you could pick from, maybe the things that indicate quality to you are how the packaging looks, if it's aesthetically pleasing, if it's clean, refined. Maybe there's really interesting marketing around it, or there's some sort of podcaster or celebrity or an influencer that's endorsed these products. Maybe that can mean, oh, well, this must be good quality because so-and-so really, champions it. Another thing that consumers think about in terms of quality is, does this product, elicit the biological effect that I'm looking for? Maybe not. And And ultimately, transparency and honesty on labels. All consumers in all areas, food, pharmaceuticals, cannabis, when we shop, we expect that the labels that are on products accurately represent what's in the bottle or in the box. So those are the things that I would say define quality from all of these different points in the supply chain. So what does poor quality cost the industry? What are the risks of not having sufficient quality controls in place? Well, regulatory gaps. If those regulations aren't tight, people ultimately can have decreased trust that the regulators that are in place are really looking out for their best interests as a consumer or as a manufacturer. Incorrect or fraudulent data is something that we hear about a lot. And ultimately, that poor quality really can lead to a negative or a poor reputation for the laboratories and for science in general. We often hear about products not matching labels. That can really hurt the brand, because if a news release comes out stating that your products have been found to not actually contain what's listed on the label, that'll hurt your business. But ultimately, poor quality, when it relates to the risk of unsafe products cause consumer harm. And that's the absolute last thing that anybody would want for the cannabis industry, especially considering that many people use these products for some sort of medical benefit. So really, all of us in the industry, regardless of where we are in the supply chain, we really have to establish expectations regarding quality and what it means to us, and potentially keep in mind too what it means to the consumer. I was actually browsing a website earlier this week for a hemp products, just checking out what was there. And I saw a product that had some very interesting marketing around it, some really interesting claims as to what the product can be used for. And when I scrolled down and looked at the reviews all said, this product didn't work. I didn't feel any different. It didn't do what it claimed that it could do. And that's really unfortunate. And that's where I think appropriate expectations are really, really critical. Because if anybody sets inappropriate expectations, it sort of sets us up for disappointment and for ultimately a lack of trust. So I am closest to the laboratory and analytical testing space. So that's kind of the hat that I will be wearing for the rest of this presentation. So if we're thinking about quality and we look at all of the products that are on the market, this is really just some examples of things that a consumer might see that are out there. None of these products that I'm showing here were actually tested in any of the studies that I'm going to present to you in the rest of these slides. Really, they're just here as an example. But if we think about quality and what that means to us, then we can better, define the question that we're trying to ask. So in terms of these cannabis products here, I would say quality. What's in these products and how much of it is there? So we have our question, and now we can work on identifying what the tools might be that we need in order to answer these questions. So I have a variety of different waters instrumentation shown on this slide that can be used for testing cannabinoids in different consumer products. So for the knowns, you can use technology that can identify and quantify the things that you expect, the cannabinoids that you know are there, and give you values that potentially could go onto labels and things like that. Or you can shift over and think about the unknowns. So you can think about what cannabinoids do I know are there that I'm aware of that exist, and look for those while also keeping your eyes out and looking and open for potentially unknowns that you may not expect. So I'm going to talk about both of these different types of technologies, but first I will start with the knowns. And with that, we will talk about liquid chromatography with UV or mass detection, and a little bit of supercritical fluid technology. So we're going to look for the knowns, right? So how much of these specific analytes are present within a cannabis product? So I have a chart here that shows 18 different cannabinoids, different names, their retention times, things like that. Waters has developed a method that can separate out and quantify each of these 18 cannabinoids in various consumer products. But on the left side of your screen, you see some molecules with some cannabinoids that were, typically more predominant or more popular in the market. CBD, THC, CBN as well. A lot of people focus on these specific cannabinoids because they have some sort of value to them. Potentially they have biological effects that people are looking for. So while there is value in quantifying and looking at these, 6, I would say, cannabinoids, there's a lot of value in also looking at a broader list. Seems like every year there's another cannabinoid that gets discovered to have some sort of really interesting biological effect. And it would be really advantageous for the laboratories to have a method that has the ability to detect and quantify a wide variety so that nobody has to go back and do method development after the fact. You already have data and a method that can meet a broad set of needs right from the start. So if we want to determine what are the known cannabinoids and how much of them are there in my products, you can follow a workflow that looks like this. So ultimately, you would take your product, you would homogenize it in some way, whether you're using, a coffee grinder, mortar and pestle maybe, or, technologies like the Geno grinder or the freezer mill to really grind your samples up into a powder that you can then go on and extract and get into solution. Once they're in solution, you may perform a variety of different sample preparations, maybe have some catcher's salts in there so that you can really separate out the sugars and have the cannabinoids in a layer that you can test more effectively, maybe filter it. And Waters also has an application success guide or an SOP that can guide people who use our instrumentation how to go about each of these steps to quantify cannabinoids and extract them and get them in the solution. And then once they're there, then you would inject them onto some sort of chromatographic instrument that will separate out all the cannabinoids chromatographically and allow you to quantify each of those individually. So one of the unique, I would say, benefits of the solution that Waters has is that we've chosen to include mass spectrometry into the mix. So usually, and most common, people use some sort of liquid chromatography with UV detection to quantify cannabinoids, right? This is an example of one of our systems that are shown here in the columns that we use. And then you can see that there are some chromatograms over on the right side of the page. Each of those peaks represent a different cannabinoid, and each of them are using a slightly different way to detect them. Whereas the top chromatogram shows the UV spectra, and the middle and the bottom actually show what those peaks look like when you're finding and detecting them through mass spectrometry. It's not always necessary because we commonly see cannabinoids that are in higher abundance, and that's usually the ones that people are looking for. But having mass spectrometry gives you almost like an additional piece of information. You would have retention time, and you would have UV absorbance as a way to know that this is the cannabinoid that I'm looking for. But mass spec is like another type of chemical fingerprint that you can look at so that you're totally confident that what you're seeing is what you think it is. One of the other cool features, or I would say enhancements of using mass spectrometry when you're testing for cannabinoids, is that you actually have a little bit of extra sensitivity. So for example, some of these minor cannabinoids are, minor, they're in lower abundance, but they might be present in a variety of different cannabis extracts that somebody might be manufacturing and producing. And sometimes UV just isn't sensitive enough to really quantify those lower level analytes that are in less abundance. So mass spectrometry actually gives you an extra order of magnitude lower detection so that maybe you're better able to quantify some of those low level analytes. It's not required, but it can add some extra value to your analyses if you're looking to quantify some of the lower levels. So one of the ways that this technology is used is for quality control testing. Some manufacturers have it in-house. It's definitely the go-to at third-party testing labs. And you get data on, cannabinoid content that can go on to be, included on different labels. But you can also use this technology to do a bit of market surveillance or even like market research. So we've heard commonly that products have been shown to not match their labels. So in this chart here, I have a variety of different cannabinoids listed, all 18, and then some quantitative data along the way for different types of chocolates and different gummies. So in the little box that popped up, you can see I've highlighted delta-9 THC in a variety of chocolates. And the third number in the white boxes after the parentheses, you'll see that's actually the percent of label claim. So for these Delta-9 products, they were a little bit over 100% of the label claims. That means they had a little bit extra Delta-9 THC in them. These numbers aren't terrible. They might be within sort of the error of a method, but it's something to be aware of, certainly, as you're formulating products. You would at least hope that the products have at least minimum what's listed on the label. Now, when we move into the gummies, quantifying those against their label claims, we've got data all across the board. So for one gummy, there was only 36% of the label claim actually in the gummy. One had over the label claim and another one was a little bit less. So it's something to be aware of. Waters did this study a couple of years ago. We weren't the first, but there were also similar observations that were published by some FDA CIFSAN researchers that reflected these results as well. And you can actually check that out in the paper that I have listed below. So why don't the products match their labels? Well, for a variety of reasons that I've shown on this slide here. Maybe there's errors in sample prep because it is sort of a finicky process. Product instability, I think, is really worth considering. Stability studies and shelf life is something that I think really needs to be considered because as products sit on the shelves, they degrade, right? Especially if they have exposure to light or if they're maybe in a little bit of a warm environment. So maybe that's why, or that perhaps inappropriate methods are used. So I won't belabor the point too much, but I did want to show that there is a really interesting paper, blog post publication online that was written by Infinite Cal and Anresco Labs that came out last year that really dives into the logistics of how is inflated THC really harming the industry and how do people actually do it. And they did this in partnership with the American Council of Independent Laboratories. It's a great organization for laboratories to come together and to discuss best practices. So I highly suggest checking that out if you want to learn more. So if labels not matching the products that are in them was, enough of a problem, we also have been dealing with the rise of Delta-8 THC products. They've certainly skyrocketed in states where there is not a legal adult use cannabis, framework, because Delta-8 products have some intoxicating properties, and you can't really miss them, and they're sold in a variety of different places. So there's a lot of access to Delta-8 products that maybe in legal states would be a little bit different. There's some harm that could be associated with using them, and there's actually just a lot of unknowns. So Waters actually did a study a handful of years ago where our researchers in-house subjected CBD oil to acid and heat treatment and then tested them to see how those products or how the chemical composition of those products changed. So in the first chromatogram on the left side, you can see those are standards. Those are known analytes, right? The second chromatogram is when CBD oil was exposed to heat treatment. And you can see that, yeah, CBD is a large peak there. It's predominant. There's some other little small peaks that are present. They actually match the untreated CBD chromatogram that's down at the bottom. So the heat treatment didn't do too much, at least that we could detect here. But when CBD was exposed to acid and heat in the third chromatogram, you can now see that there are a variety of different peaks that suddenly appear within this chromatogram. One of which was definitely Delta-8. And there were a variety of other, analytes that showed up because you're performing organic chemistry. You're doing synthetic conversions using chemistry. So that's a little concerning. Well, Delta-8 products are, created from CBD by some, chemical transformation. We did this on a small scale, but on a large scale, that shows that there are some potential, new things that are popping up in these extracts that we should probably characterize and know a little bit more about. So how do we go about doing that? Well, that's when you can go and move towards some of the more advanced technology, including high-res mass spec and ion mobility, so that you can look for the cannabinoids while also gaining some extra information about those unknowns. So here I am showing a chromatogram of some standards and of a Delta 8 THC distillate product that was provided to us by one of our collaborators, something that was going out on the market. So you can see that every peak in these chromatograms that are labeled with a number are knowns. We can see that in the chart. Like 1 is CBD, 6 is CBL, 4 is Delta 8 THC. We know the retention time of those analytes. And we also know that they have a mass to charge ratio of 315. Well, if we go and look at the distillate sample, the bottom chromatogram, you can see that there's some peaks there that are labeled with numbers. We know what those analytes are, but then everything that's labeled with a letter is an unknown. What are these unknowns? How do we go about discovering what these are so that we can better understand if these products are safe for consumption? So I'm going to zoom in on this section of the chromatogram that shows peaks A, B, C, and D. So if we take this Delta 8 THC distillate sample and analyze it using liquid chromatography and high resolution mass spectrometry, we get additional information. With high resolution mass spectrometry, we actually are able to have a more precise measurement of the accurate mass of these unknowns, so we can better zone in on what the potential chemical formula is. We can also fragment it into pieces, blast it up so that those puzzle pieces that are, generated from fragmentation can be sort of put back together in a way to hypothesize what the potential structure could be. So we've got retention time, accurate mass, fragmentation data, and with that, in our software, this was tested using our new Xevo G3 QTOF on our Unify software platform. We can take all of this data and feed it into databases and say, or get some predictions of what these unknown analytes might be. So peak D was what we zoned in on, and we found that in sort of the middle right of the slide, there are some potential component names. Those are predicted analyte IDs. So now that we have maybe some predictions of what this unknown D might be based on its chemical features, we can search the literature and say, do we know anything about these compounds yet? Is there any safety data that's associated with it, any sort of clinical studies? And from there, we can say, all right, well, maybe this is less of a concern, or maybe it is something that needs to be further addressed. So based upon our testing using the Xevo G3 QTOF mass spectrometer, high-res mass, we were able to predict that this is unknown D is a hexahydrocannabinol isomer. And here's just an example of some of the publications that came out that mention this isomer, that sort of have some maybe pharmacological data about it. This is a step in the right direction towards better understanding what's in these products. But we can take it to another level. And it gets a little bit more complicated. So I'm showing again that chromatogram of the distillate sample. And one thing I want to show you is that sometimes one peak, one spot in a chromatogram, it might look like one peak. That doesn't always mean that it's just one analyte represented there. So we tested this sample using our Water Select Series cyclic IMS, ion mobility mass spectrometry, and it adds a different dimension of separation to whatever analytes might be present in your sample. And the way it does that is through a different dimension of separation, ion mobility. And I think the easiest way to show you how ion mobility works, because I could talk for 2 hours as an introduction, is really demonstrated well by this little, graphic that's going to pop up. So ultimately, you can have 2 pieces of paper or 2 molecules with the same mass, and you can change the shape. Maybe they, exist in different 3-dimensional configurations. And if you move them through air, the one that's more, expanded out will travel slower, will drop to the floor slower than one that's a little bit more condensed, a little bit more tightly bound, right? That is essentially how ion mobility works. You separate things out based upon their 3-dimensional, positions in space. The way the cyclic does this is it's this kind of like racetrack setup where you can send those ions and analytes into this racetrack. It spins around, separates things out in space for theoretically infinite passes. And ultimately, you're able to separate out maybe things that are almost identical, but slightly different. So we did this using this system on this distillate sample, and we found that depending on the number of passes or laps that you take on that racetrack, what was once one peak was actually two overlapping compounds with the same mass, the same retention time, and the same chemical formula. So here is a different view of the data from this instrument. You can see that we have peak E, F, G, and 2. 2 is a known, remember? Letters are the unknowns. So if we take this section of the chromatogram, we're analyzing these samples, and we only do one pass through the racetrack, we see essentially 4 peaks. Now, if we do 3 passes through our ion mobility racetrack, you can start to see that those peaks are kind of blobbing out, spreading out a little bit. That means that there was more than one analyte underneath that sort of chromatographic space. And we never knew that until we did ion mobility. So you can see that F actually had 2 things underneath it, and 2 actually had 2 things underneath it. And you can see it even better when you do 6 passes through the ion mobility racetrack. So that can be a little bit alarming. Sometimes the more you know, the more you wish you didn't know, right? But now we know that there are even more compounds within this, distillate sample that need a little bit more, testing, or maybe a little bit more work to identify what they are and if they are a hazard. So we're asking ourselves now, what are these compounds? What are they? Do we know what they are? Are they a concern? So the one additional cool feature of this instrument and this, advanced technology that allows you to better understand the unknowns is that you can separate things out within that racetrack, and you can actually selectively send those ions into that fragmentation cell, blast them up into pieces individually, which you never could do before. So you get an even closer, level of detail that can help you determine what these unknowns are, because they fragment slightly differently. So in the right side of this PowerPoint slide, you can see that 315 is the largest peak that's there, but some of these little minor, peaks that are really hard to see, but they're labeled with numbers, are a little bit different. And that helps researchers to know maybe things are put together a little bit differently between these 2 molecules. So they might be pretty darn close to each other, but they are slightly different. And sometimes those slight differences can make an enormous impact on human health. So now what? That is the end of my scientific story and what we have to present at Waters. More so a selection of tools that can help a person better understand what's in these products. Our role is that we create these advanced technologies and then put them into the hands of researchers and developers so that they can really take this instrumentation, run with it, and unlock some of these really hard scientific questions. So, here's just another selection of publications that have really dove in to figure out what are some of these impurities, these unknowns within these delta-8 THC products, and are they something that we should be concerned about? So there were 2 publications in October, and essentially hot off the press within the past month, there was another publication in the Journal of Natural Products by the El Soli Research Group at the University of Mississippi that went on to further characterize some of the impurities in commercially marketed and sold delta-8 THC products. More research is needed. There's more happening every day. There's always something new to learn. And ultimately, it's all about protecting the consumer and the industry. So I asked at the beginning of this presentation is how do you define quality? How do we know if these products are high quality? Well, it ultimately depends on what quality means to you. I would say that having products that aren't fully chemically understood can cause me to pause. And I think it causes many others to pause as well. It's just something to be aware of when we all consider our role in the supply chain. So how can the analytical labs champion quality within the cannabis industry? Well, defining what quality means and using fit-for-purpose methods is a great way for analytical labs to make sure that their data are the highest quality to support the different products that they test. And analytical labs, if they want to ensure that their service, their testing methods, and their data are the highest quality, I think routine participation in proficiency testing programs is really important. It's essentially a test for the labs to say that their methods are working as they need. And there's a variety of different providers, Emerald Scientific, NIST, AOAC International, that have different types of cannabis proficiency testing programs that the analytical lab industry can really, engage in. If you're here and listening today, or if you're at any of these conferences that relate to cannabis science or botanical science, you're actively engaging in contributing to the scientific community. I know that there's a certain level of, brand privacy that many like to, have, including some of maybe the methods that they use in order to accomplish their goals. But if you're able to contribute to the scientific community and share your challenges and your successes, it'll only bring the entire industry up. And ultimately, speak up for quality. There's a lot of stuff happening in the cannabis space. I feel like we often hear more about the bad than the good. So I challenge everybody here to really speak up for quality and engage in the scientific community and the conversation and share how you can, so that we can continue to just really bring up the level of science to where it needs to be for this product. There are a few different ways that you can get involved. I mentioned the American Council of Independent Laboratories as one. USP, ASTM both have stakeholder groups that really work together to improve the understanding of cannabis and the safety of consumers and those who work in the industry. And AOAC International is another organization that's really championing fit-for-purpose methodologies. So that is where my presentation will wrap up. I do have to say a special thank you to ProVerde Laboratories. They're a longtime collaborator with Waters. We have a lot that we can thank Chris Hidala for and Andy Aubin for. Both of them are at ProVerde. Chris is actually one of the co-founders of ProVerde. So we are very thankful for their collaboration and their ability to share samples with us. I also want to thank Marion Tuhig, Catherine Layton, and Kim Tran, 3 fantastic scientists at Waters Corporation that were actually the ones who had their hands on the samples and were doing the testing of all of the data that I presented here today. I'm very fortunate that I get to be a messenger of the incredible science that they do. And then I also want to thank AOAC International, ACIL, and USP for just being incredible, venues for collaboration and learning. If you're not involved in any of those today, I would say sign up. So thank you all so much for listening to my presentation today. I appreciate every moment that you spent here. And if you have any questions, you can add them into the chat box or get a hold of me on LinkedIn or by email, and I'd be happy to answer any of your questions or provide any assistance that is needed. So thank you all. I hope you have a great day and enjoy the rest of the Lab Roots event.